offering ourselves at the founder chancellor of Sri Satya Institute of Higher Learning, Bhagavan Sri Satya Baba. I welcome you all to this popular science talk series organized by Center for Excellence in Mathematical Biology, which is stationed at Sri Satya Institute of Higher Learning. So before starting the first talk, I just thought maybe a couple of minutes to go take you a virtual tour through the Center for Excellence in Mathematical Biology. So this is part of this popular science talk series, what we are doing. So we are also started organizing faculty development program. This is in pipeline. We just released the flyer yesterday. Faculty development program on dynamical systems and optimal control theory, AML and bioinformatics. So when you click the web link, ssihl.edu.in and CMV, you can see the details when you click this hyperlink. So this is quickly a recap of the vision statement of our Center for Excellence. We are more focused towards practical applications of whatever research has been done. And here is a quick bird's eye view executive summary and the aims and objectives of the center. So before going through the members presently as chief patrons, we have the vice chancellor and the registrar. Then of course I am the coordinator for this center. Then we have national scientific advisors from the reputed institutions spread across the country, and also we have scientific advisors international. So these are some of the, here are some of the core committee members. As you can see, the center has members which are spread across the different parts of the India from different reputed institutes. And yeah, recently those are the core committee members who contribute to whatever activities we do. And here are some of the members from SSIHL and few other institutions. So coming to the activities, this is in fact a center that was started very recently. In the last November 22nd, our revered vice chancellor has announced the opening of this center. So since its opening, we were active. So we have organized a couple of workshops in infectious disease modeling, and also, we majorly on the infectious disease modeling at Lovely Professional University and ICMR and IIRT. And also we organized in the field of mathematical biology, a couple of conferences. One is the national conference, other one is the US Indo conference. And all of you know, I think most of you might know about the infectious disease modeling certificate program that we have been offering. So not taking much of your time, here are some of the objectives. We work on TB, cancer, Hansen's psychological modeling, as well as graph theory. And also these are some of the center focuses primarily on the research and innovation, the education aspect. We offer specialization in mathematical biology. Of course, this popular talks is also part of the research and innovation and the extension, outreach activities. These are some of the contacts. So we are yet to update the website. So we have also recently started collaborating with our sister institutions, Sri Satya Institute of Higher Medical Sciences. So we have started collaborating. I'm happy that the joint director of SHIMS, Prashanti Gram, is also with us. So we are working on cardiovascular disease, especially in the data analytics and the AML part of it. So that's also part of the proposed faculty development program that we are organizing in July. It's going to begin from 8th and also end on 15th. So this is in brief about the center. So I would urge all of you who are in the meet to kindly go through this Satya Institute of Higher Learning CMB website. So, and if you are interested, you can always join us and we can do collaborative work. So that's in brief about this center. Over to Dr. Darshan for introducing the speaker for today, Dr. Mohit Jolly. Yeah. Over to you, Dr. Darshan.
Yeah, thank you, Dr. Vamsi, for taking us through the center. Now I'll just share my screen. Is it visible?
Yeah, please go ahead.
Yeah, okay. So good afternoon, everyone, and a warm welcome to everyone. Now it is my pleasure to welcome you all to this first session of Popular Science Talk series organized by the Center for Excellence in Mathematical Biology at Sri Satya Sai Institute of Higher Learning. Today we are honored to have with us Dr. Mohit, an associate professor from Bioengineering Department at Indian Institute of Science, Bangalore. Dr. Mohit is a renowned researcher whose work combining computer mathematical models and cancer biology has gained worldwide recognition. Dr. Mohit completed his bachelor's and master's at IIT Kanpur and then earned his PhD from Rice University, Texas, USA, all in the field of bioengineering. After joining Indian Institute of Science in 2018, Dr. Mohit has led pioneering research on the physics of cancer, focusing on developing computational models which untangle the complexities of cancer progression. Dr. Mohit's innovative work has been recognized with prestigious awards such as the ICTP Prize 2023, IIT Kanpur Young Alumni Award in 2022, and most recently, Dr. Mohit was honored with Rice University Outstanding Young Alumni Award in 2024. Dr. Mohit also serves as the editor-in-chief at NPJ System Biology and Applications. In today's talk, which is titled as What Does Not Kill Cancer Can Make It Stronger, Investigating Cancer as a Complex System, Dr. Mohit will explore the challenges of cancer metastasis and therapy adaptation. Now, with these few words, I welcome Dr. Mohit to share his groundbreaking research and its implications for advancing the cancer treatment. Dr. Mohit.
Yeah, Dr. Darshan, can you unshare your screen so that I can
Yes.
share the slides? Yeah, just before Mohit sir, just let me know if everything is going fine. So I just share the slides. So can you see, just a second? Can you see the slides? Can someone confirm? Yeah.
Can you make
So,
it full screen if it is possible?
Yeah, I made it full screen now. Can you see it? It's in presentation mode.
Yes, yeah, we can start.
Yeah. So, Dr. Mohit, request you to kindly tell me to change the slides so that I can.
Sure. Thank you. Thank you. Thank you very much, Dr. Vamsi and Dr. Darshan, for this very kind invitation. It's my real pleasure and honor to share some of our work here at this popular science series. And it was a pleasure visiting GSAT Science Institute of Higher Learning last July for an exciting workshop, which was also put together by Dr. Vamsi and other colleagues. And I'm really glad to continue our discussion and potential collaborations as well. So what I'll do in the next 40 minutes or so is walk you through the how mathematical models can be used and examples of how mathematical models can be used to understand the dynamics of specific processes in cancer, which happen to be the most challenging scenarios in the clinic, which is how cancer cells metastasize, that is, move from one organ to another, and the other challenge being that how they develop resistance to different drugs that are being currently used in the clinic. Next slide, please. So before we go into details, I want to first talk about what exactly we understand by cancer. So cancer is referred to as an emperor of all maladies. It is not a disease, it is actually a set of diseases, each disease showing up at different organs, and even cancer in a different organ can show up in different subtypes as clinically defined. So the basic properties of cancer, the two most well-defined properties of cancer are that cancer cells divide in an uncontrolled manner as compared to normal cells in our body, which have an intrinsic program that somehow controls how many times they can divide during their lifetime, and therefore when cancer cells start to grow in an uncontrolled manner, that disrupts the organ architecture, function, and that is what eventually leads to patient death. So that is one defining feature about cancer. The other defining feature about cancer is that it can, if you think about solid tumors, they can actually move from one organ to another. So you might have heard of, say, breast cancer cells actually showing up in lung, developing lung metastasis or liver metastasis, prostate cancer patients often having metastasis in the bone, and so on and so forth. So these are two defining traits of cancer. Most other diseases that we know of actually do not start spreading to different organs. They, of course, have physiological implications that transcend the original organ where they're initiated in and have impacts on the anatomy, but they do not dislodge themselves from one place and start going to other places. Next slide, please. So these are two unique features which are specific to cancer. Next, please. And here is an image which actually shows the pancreatic cancer cells growing in the liver, the image that you saw, where you could see that in a liver, the entire architecture, there were many of these nodules, which were basically pancreatic cells that have actually come here. Next slide, please. Now, if you think about what are the total numbers, how many patients, how many people cancer impact? So these are numbers from the US over past 50 years approximately. So what you see is that by say 2040, the expected number of cases, the projected number of cases is somewhere around 25 million. And in the US, next slide, please, every second cancer death in men and every third cancer death in women is actually attributed to cancer. And number of cases in India have also been growing. We are expected to have more than 15 lakh cases next year. Next slide, please. Now, India already had about 10 lakh cancer deaths. This is 2019 data. And if you look at the US data on the graph on top, the number of deaths, they have not come down, but they have also not drastically increased in the past 20 years in the US, and those numbers are around 6 lakh. So we as a country are already having more number of deaths due to cancer as compared to the US, despite having much less number of cases. This is of course happening due to various reasons, including late detection, including the different cancer constitutions. We have, for instance, in the breast cancer scenario, the incidence of a more aggressive subtype of breast cancer have been seen clinically in India as compared to US, the reasons of which are still being investigated. But that's where the numbers are. Next slide, please. So what you see, as I was mentioning to you earlier, is that in females, 1 in every 3 and 1 in every 2 men, they actually get cancers. That's the US data. And in India, we are not that far behind. We, as per an ICMR report, somewhere around 1 in 9 people are likely to develop cancer during their lifetime. Next slide, please. So when we think about this disease, what are the current treatment strategies that we have for this disease, right? So the earliest strategy that has been adopted for a long period of time now is surgery, where, for instance, in the case of breast cancer, the example that you see here, which is that you remove the organ as much as possible. So the mastectomy being done here, which is basically removal of the breast. Then this was, of course, earlier in the day. More recently, there are other options available as radiation therapy, which came around the turn of the previous century, when it was identified that X-rays can have such an impact on killing cells as well. And around the time of the World War, when chemotherapy came in, when it was realized that certain gases which were actually being used for different warfare can have an impact in terms of killing specific cells. Around 1980s, a more specific therapy, which basically means if you compare it to, say, surgery, radiation, or chemotherapy, these are more generic therapies. Now, in targeted therapy, which is that you identify a vulnerability which is unique to a cancer cell, or largely unique to a cancer cell, and then develop drugs against it so that you minimize the side effects which are happening because of these therapies. Say, for instance, chemotherapy. The general principle is that it kills fast dividing cells. Now, there are many fast dividing cells in the body, not just cancer cells. Hair is one of the fastest growing organ, and that is why you see many patients receiving chemotherapy actually have less hair, especially the head hair, and they get bald during, because so that's a direct consequence of chemotherapy itself. And immunotherapy, which is the latest therapy that we have. In this case, we do not directly manure the cancer cells, but we actually overactivate the immune cells. And in that case, what happens is that the immune cells can then go and kill the cancer cells. So this is the latest scenario that we have here. Next slide, please. So we have all these different options for this disease, but we still have not conquered it, as one would say that we have, say, largely conquered different forms of cardiovascular diseases. And of course, I know of the excellent work being done at Rishi Sathya Institute of Medical Sciences as well in this field, and also as Dr. Wangsi was mentioning about the collaboration, right? So let's go a little deeper into what the different stages of cancer progression are. Next slide, please. Now, in this case, here is in a schematic, what you see is that cancer is initially, say, initiated in one or more cells due to various disruptions, and that cell, because now it has the ability to grow much faster and much more than the normal cells shown in pink, you see that these cells begin to show up much more prominently and then disrupt the organ architecture and then go to other organs as well, which is what is the process of metastasis. Now, the timeline that you see can, of course, vary depending on which cancer and which patient we are talking about. But the general point I'm trying to make here is the acceleration that happens once cancer begins to spread. The initial phases are relatively slower in terms of the mass being showing up on in a given patient. Now, over the past few decades, we have made some excellent progress in diagnosing cancer early. So now there are tests for both men and women to get tested, at least for breast cancer and prostate cancer likelihood. There are also progress being made in terms of what genes in a given cell are accelerating or deaccelerating the process of cancer, what lifestyle factors such as smoking causing lung cancer has been very distinctly shown. But as you would see, that all these factors largely restrict themselves to initial phases of cancer. Once cancer metastasizes, we have not really had much more breakthroughs in controlling it in that particular stage of the disease. Next slide, please. So then the question comes that why we have not been able to, next slide please, yet understand the dynamics of metastasis, or why have not been able to control metastasis. Next slide please. So metastasis still remains the cause of all cancer, a large proportion of cancer deaths, and the way cancer cells spread from one organ to another is largely using this fantastic freeway of blood vessels that we have in our spread all over the body. So cancer cells in one organ actually get on this freeway, travel to different parts of the body, and then they can actually begin to grow in different parts. Now, next slide, please. The main point about when I started thinking about metastasis, the dynamics of this process, which struck me was that if you think about the most common cancers, the solid tumors I'm talking about, say breast cancer, prostate cancer, lung cancer, liver cancer, etc., they do not have the intrinsic property to move. They are actually stuck in a specific three-dimensional geometry, very tight cell-cell adhesion with their neighbors, and also segregated to form different structures, which actually gives them function. So how is that cancer cells then begin to move? And once they have moved, how do they know that they have to stop at a distant site, right? And that lack of understanding of the dynamics of this process is actually a major cause why we still do not have drugs against this, right? So let me draw an analogy. So the first picture that you see on this slide is close to the first scene in the first movie of Harry Potter series, and the last picture that you see on this slide is close to the last scene in the last series of movies in Harry Potter. Now, if I were to give you only these two images, and you can replace it with any other two images of any other movie series or TV series and so on, and just ask you, just by giving these two images, tell me what happens in between. It's impossible to tell what the entire sequence of event, in what sequence it happens, such that you go from point A to point B. That is where we are, largely speaking, when it comes to metastasis research. We have patient sample from primary tumor, and finally we have patient sample from metastatic tumor when the patient comes back after multiple months or years back to the clinic, and cancer has shown up at a different organ, right? So there is very limited understanding that we still have as a field, although we have made great progress in the past 20 years, I would say, but still many of these questions we only know in preclinical settings, such as mouse models, for example, or laboratory research, which is that when and why or how do cancer cells begin to leave the primary tumor? Do they leave as individual cells? Do they leave as groups? Does it matter whether they leave as individual cells or groups? If groups, then what is the typical size of a cancer cell cluster that is leaving the primary tumor? Do they actually stop by different organs, or do they only go to specific organs in the very beginning itself, and how much time it takes for them to grow a metastasis, right? So these are of course complex questions. These are not easy questions to understand, and as I said, we have some answers to some of these questions based on preclinical research, but that level of understanding has not advanced so much that we can actually develop drugs against this entire process. Next slide, please. Now, the other challenge is that of drug resistance. So for instance, again, in this example, which is that of a patient, what you see is that, so this is a patient of melanoma, which is skin cancer. So pre-treatment, you could see all these different nodules which are present on the patient upper body. And then 15 weeks into the treatment, you could see that these nodules have disappeared. So the therapy that is being given to this individual has actually helped the tumor mass shrink dramatically. But only 8 weeks later, in presence of the therapy, what you again see is that nodules reappear. Now, what that tells us is that clearly at 15 weeks, when we are seeing no discernible sign of the disease, there are some cells which are left behind, and those are the ones which are eventually able to grow this entire tumor again in a short duration of 8 weeks. Right? So this is what is called as minimal residual disease in a patient, or the phenomena of drug resistance. So again, there are these questions which we have some exciting answers in the past 10 years thanks to the advancements in development of technology. But when we will reach such an understanding that we can use these answers to develop new therapies in the clinic, that is a process that is still ongoing. So were there cells, these pre-existing cells, present even before the treatment was given, or did the drug treatment push some of the cells to acquire this state? What controls the time scale? Why 8 weeks? Why not 8 months? How does different patient or the therapy given controls these timescales? All these are exciting questions for which data is beginning to accumulate, such that mathematical models can now play a big role in understanding the fundamental rules and taking the research forward. Next slide, please. So now if we think about the dynamics of this process a little bit more quantitatively, let's say that 10,000 cells leave a primary tumor. And how many of them will complete metastasis? If anyone wants to take a guess, please answer it in the chat. Is this be somewhere between 100 to 1,000, between 1,000 to 10,000, between 10 to 100, between 1 to 10? I'll just pause for maybe a few seconds to see if anyone wants to guess what these numbers look like in. Okay, so 50%. So that's one answer. Anyone else? Anyone? 5%. Okay. Yeah, so you could clearly see the answers are strongly varying. And when I say complete metastasis, I mean that they would actually grow a tumor, which is notable in the clinic. Okay, so the answer is 1% or less. So it's a highly inefficient process, and thankfully so, because otherwise you would see much more metastasis in patients. And actually, next slide, please. One of the experiments done around 25 years ago gave some answers to this. So this experiment was, of course, from a mouse model. What was done was that in the vein that goes to the liver, some cancer cells were injected. Now, of course, the experiment has this limitation that you are putting the cells in the vein directly. So you are skipping one step, which is that cancer cells getting onto the bloodstream. So the actual process is perhaps even less efficient than this. But nonetheless, even if you skip the first step and you look at how many cells actually grow what is called micrometastasis, which is only 4 to 16 cells, so a cell dividing 2 to 4 times, and then how many actually grow a tumor, the probability is actually very low. So what is so special about that minuscule percentage of cells which is actually able to grow, which is able to metastasize and grow a tumor at different sites? Next slide, please. And this is where what Charles Darwin said becomes super relevant, that it is not the most intelligent species, it is actually the most adaptable species, which is eventually going to make an impact, right? So if you think about the journey of metastasis, then these cells are facing all these different challenges, and there are very few cells which are able to complete or passing through all these bottlenecks in a successful manner. So these are challenges because of lack of oxygen and nutrients, external therapies, shear stress, ability to adjust adhesion, migration, etc. All of these properties, right? So if you compare this dynamics to some of you who know of or might be thinking of, say, going to a completely different foreign country and settling down there, one of the key properties to succeed in that process is that of adaptability, right? And that is what years of cancer research has now shown, that it's adaptability that is a feature that dictates the success of metastasis. Next slide, please. Next slide. Now, the next question that comes is that, is every cancer cell equally likely to adapt or equally likely to metastasize, right? And the obvious answer is no. And this was again done in a very elegant experiment, which was again done almost 50 years ago, which actually showed that not every cancer cell is equally capable of metastasis. So what was done in this experiment is that if you take a cell population, which is the graphs on the left hand side, so if you take, say, a large number of cells and then inject them in mice and see how many tumors they form, so from multiple readings you'll have some distribution. And now, instead of taking the whole population of cells, you grow the population from a single cell. And from that single cell, then you repeat it to the same observation, that how many cells are actually able to grow tumors in a mice. And the answer is, as you can see in the right bottom panel, is actually highly varying. In some of them, they were able to grow many more metastases as compared to others, right? So just in any distribution, of course, the numbers are not going to be all centered around the mean. There are numbers which are going to be much larger or much less than the mean. So even if you were think of adaptability as a function of as some f of x, then clearly there's a distribution here, and that is was very neatly shown in this experiment. Next slide, please. So what we know so far is that cancer cells are heterogeneous, that there are many cancer cells, many different types of cancer cells in a tumor. Not everyone is identical, and they also have this ability to adapt. And these are, for technical terms, called phenotypic heterogeneity and phenotypic plasticity, that they have different personalities if you draw analogies to human behavior, and that personality is also a dynamic function which can change as a consequence of different interactions going on within the community or even within a cell. Next slide, please. So now let's look at the dynamics of tumor relapse, right? So this is again the example that I presented to you from a patient of melanoma. So this is a mouse model or the preclinical setting of the same tumor and given the same drug. So what you see on the top left panel is that initially when the drug is given for about 15 days, there is a reduction in the tumor volume. And for almost a month, the tumor volume does not increase, and the drug is constantly being given, but then the tumor volume begins to increase after that. Right? So what is going on in that period of shown by the yellow region, light yellow region of adaptation, is that cancer cells are switching to a different behavior, which somehow is increasing their fitness in this environment, and that is allowing them to grow eventually, right? And this is what is called as minimal residual disease, as I was referring to earlier. So what they're doing is they're changing their cell state, and there's a transition that is happening from cell state 1 to cell state 2, and none of the therapies that we have right now, which we talked about earlier, actually accounts for these cell state transitions, right? Surgery, chemotherapy, radiation therapy, they all think about one specific feature of cancer, and they do not explicitly account the possibility that cells can actually switch their behavior and take up a completely different phenotype. Next slide, please. So, for example, in this case, that let's say if you have a tumor and you have mapped the tumor as extensively as you can, and you have, say, 4 different types of cancer cells, and you also have targeted therapy against each of those different 4 types, you still have so many different assumptions that are ongoing here when you think of the current therapies, right? So first of all, we are assuming there are only 4 cell types. We are assuming that each red cell in this population will respond identically to a given concentration of that drug, and will have one and only one outcome, which is death. Right? That's a big assumption to make. We are also assuming that the drug is not able to induce a switch from one state to another state or give rise to new states which were not seen in the prior population. And we are also assuming that there is no cell-cell cooperation going on when the population is facing stress, right? So all of the therapies that we have now are basically implicitly assuming that cancer is a static system, and non-adaptive system, but cancer has been shown both in based on clinical observations and preclinical studies that have been going on in the past at least 30 years or so, has shown that that's not the case. Cancer is a very dynamic and complex system, and we need to begin to appreciate it, and that's where the role of mathematical models become super important. Next slide, please. So then the question comes, how can a mathematical model help? And I don't need to talk to this audience about in much more detail. These are tools where you could actually take different interactions with a system and understand the emergent dynamics, go into detail of how individual factors are actually changing the dynamics. Next slide, please. So the question we started thinking about was, can we develop a mathematical model to study metastasis and drug resistance? So again, before I go into details, there's a quick poll here. How many of you developed a mathematical model in past 1 week? Past 1 week, past 1 year, or never? Type in A, B, and C in the chat. Okay, people who are saying never, okay, we will get to that. I'm sure you have, and I'll just present some examples when you have developed a mental model but may not have necessarily realized it. Okay, next slide, please. So any of you who has played any sports have developed a mathematical model, trust me. You're developing it mentally. You are not necessarily writing it down and using computers to solve it, but you are using the laws of physics or laws of motion to actually control your behavior and make sure that you hit the ball in a specific direction with a specific velocity, etc. Any of you who has driven a vehicle and made estimates whether they'll be able to cross the light before it turns red or not, for example, has developed a mathematical model. Again, these, as I said, you're not explicitly writing it, but you are using the laws of dynamics to better understand it. Anyone investing in stock markets, etc., and so on. Right? So we all develop different kinds of models. Some are more formal models, some are more mental models to answer specific, precise questions. Next slide, please. And the question we wanted to address here was that, can metastasis and drug resistance drive each other? Can these processes actually influence each other or accelerate each other or amplify each other? And this was coming from this observation, clinical observation, that 1/3 of the women treated with this drug called tamoxifen, which is targeted therapy for a specific subtype of breast cancer, for 5 years, they actually have the disease come back in about 15 years, or within 15 years, and many of them have metastasized meanwhile. So how is drug resistance and drug resistance and metastasis happening simultaneously? And this is a clinical graph that you see where the x-axis is years, and the y-axis is the rate of occurrence at a different organ than breast cancer, than breast itself. And the three different lines actually show the different clinical stages at which the disease was diagnosed. Next slide, please. Now let's think about, learn a little bit about what the different breast cancer subtypes are. So clinically, when a breast cancer patient has to be categorized in terms of what treatment to be given, the pathologist usually looks at the levels of these markers that are mentioned here. These are surface molecules present on the surface of a cell called estrogen receptor, progesterone receptor, or ER or PR in short, and HER2, which is yet another receptor. Now, some patients do not have these three markers present to the extent that is expected in a cancer. So this is what is called as triple negative breast cancer. Now, ER positive breast cancer is the most common type of subtype of breast cancer. And what happens in this is, so ER as the name suggests, responds to estrogen, which is a growth hormone present in the environment. And it is also present in a normal breast cell, but in a cancerous breast cell, they are present to a much higher degree. And that increases their sensitivity to estrogen being present, and that leads to much faster growth. So what targeted therapy, or tamoxifen in this case, does, next slide please, is that it actually binds to estrogen receptor and prevents its binding to estrogen. So now, even if there are more number of ER molecules on a cancer cell as compared to a normal cell, but many of them are rendered useless because tamoxifen is able to bind to them and they are not able to grow as fast. Right? Now, you could think of various different reasons why this therapy may not work. Not every cell necessarily has such high levels of estrogen receptor. There can be structural changes such that tamoxifen is not able to bind. And of course, estrogen is not the only way in which cells grow. So there are all these caveats, but nonetheless, next slide, please. If can we think of other possible reasons as well? Right. So one of the things that I mentioned to you earlier is that cancer cells, if you think of the most solid tumor scenarios, they do not have the intrinsic property to move. They actually gain the ability to move, and through this process, which is called as epithelial to mesenchymal transition, or EMT in short, which is how cancer cells begin to move, largely speaking. And once they reach the other organ, they no longer need to migrate or invade. They actually need to settle down and form tumors again. And this is what is the exact opposite phenomena of MET, or mesenchymal to epithelial. So these are two different names. Let's not worry too much about them, but the point is that they have different properties. In one case, the cells are stationary, tightly cell adherent, and the other case, cells are actually moving and not adhering to the neighbors. Next slide, please. So the question we had was that, is estrogen response, or sorry, tamoxifen response connected to EMT? So what we did is we actually went through the literature in detail and the references that you see at the bottom of the slide. And what we saw, and I'm presenting a part of our work here, we found that there was these two, even estrogen receptor has two different forms. One is which allows tamoxifen to bind to it. One, the tamoxifen is not able to bind to it due to structural changes in the way this molecule is. So, and what also is known is that these molecules can control the abundance of each other. So they're not just interacting with each other. So when the levels of ER66 is high in a cell, it triggers a whole set of biochemical reactions that represses or that reduces the cell to grow ER36 or to produce more ER36 in a cell. And that is the tamoxifen resistant version of it. And tamoxifen resistant molecule can actually trigger EMT by increasing the levels of these molecules. And again, so green is increase and red is decrease, right? So if you think about the system, there's a nice feedback loop being formed here. And what is happening is that, let's say if ER66 goes up in a cell, ER36 will go low. And because ER36 is low, EMT will be low. And because EMT is low, ER66 will continue to be high. So this is one state of the system. And the other state is the exact flipped case, where if ER66 goes low, 36 goes high, cells are more migratory, EMT goes up, and because EMT is high, 66 continues to stay low. So this is a clear bistable system that you are seeing here. In one case, cells are stationary and not responding, stationary and responding to tamoxifen. In the other case, they are migratory and not responding to the same drug, right? Next slide, please. And then we did stochastic simulations to show that actually cells can switch their cell states from state A to state B under the influence of different kinds of noise. Next slide, please. And the question we wanted to ask was, can one behavior drive the other behavior, right? So when cells are undergoing EMT, can they drive tamoxifen resistance and vice versa? And we did find examples of that. And also when we looked at publicly available data, which is what I'm showing here on the slide, you actually see that when you, on the left hand side, the panels show that when you take epithelial cells and drive EMT through multiple different molecules, twist, snail, and slug, the estrogen response comes down. So the cells are less likely to respond to tamoxifen, and vice versa. When you drive tamoxifen resistance on the right hand side plots, then you see on the rightmost side that EMT goes up, right? So these processes, there's reciprocity here. So when cells are beginning to move, they are already gaining drug resistance in that property, in that phenomena. And when cells in a tumor are being exposed to increasing stress, and therefore they are gaining resistance, eventually some of them, a small subpopulation of them, those are the ones which are also becoming more metastatic simultaneous, right? So they are both driving each other. Next slide, please. And that is what might be perhaps causing why drug resistance and metastasis happens together, because these processes can actually drive each other. So based on this, then you can actually suggest that you should be thinking about different combinations of therapies, that tamoxifen should be given together with EMT inhibitors, for instance. Next slide, please. The other quick example I'll talk about is that which I've already mentioned earlier, melanoma, tumor relapse. This example I've showed you earlier. We also have a targeted therapy for melanoma called vemurafenib, and there are many others, but this is the example I present here. Next slide, please. And what in this example, this is experimental data from other colleagues. What you see is that in the left, so these are two markers. Think of this as a scatter plot. The color is telling you the density of cells in a given quadrant. So before the drug treatment, you could clearly see that most cells are in the first quadrant. So they have both of these molecules, which is marking two different functional axes in a cell. But 10 weeks after treatment, you see that the population structure has changed. You no longer have a large proportion of cells which are there in the first quadrant. And in fact, you begin to see many cells in the fourth quadrant, which were not present prior to treatment. Now, whether the cells in first quadrant have been killed, or whether they have been switching to the fourth quadrant, or second quadrant and second quadrant switching to fourth quadrant, all of that information, all of those scenarios are possible, but this data alone cannot dissect what different scenarios have happened. But it clearly shows you that in presence of the drug, it is possible to have new cell states which were not present prior to treatment of the drug. So the drug treatment can actually be giving rise to new cell states which were not existent earlier. Next slide, please. So then the question we asked was, were these cell states existing prior, or were they pushed by the drug? And our model shows that both scenarios are possible. So we modeled a regulatory network similar to the one I showed you earlier, showed that it is multi-stable. You can have multiple states. And when you actually go and look at the real data, now this is much more refined data as compared to what you had earlier, because technology has now improved and you can look at a single cell level, which is what is shown on the right-hand panel. So when you look at the untreated cells in orange in the top plot, you see that there are two subpopulations, one which is more proliferative, so these cells are growing more but invading less, and the other which is more invasive and proliferating less. So cells are doing one thing at a time, largely speaking. But in presence of the drug shown in the blue, you see that the population highlighted has disappeared. Now, why has this disappeared? Has this switched or not? That information you cannot get alone from this plot. But if you look at the bottom plot, you get more information that when you think about combination therapies, that actually can again give rise to new cell states. So the red and green populations were not seen prior to treatment of the drug. So clearly there is switching going on. No doubts about that. Next slide, please. So if you put both these examples that I showed you together, what we and many other colleagues in the field have shown through other examples and more detailed studies of these examples, that cell death is not the only inevitable output for a therapy, right? You could have different outputs, such as the cells actually becoming more aggressive as well. And mathematical models have a crucial role to play here, is to integrate that information and then suggest better combination of therapies which can lead to minimizing these side effects potentially. Next slide, please. So then the question comes, why do you need mathematical models? You could take any two drugs which are approved by, say, the Food and Drug Administration, FDA, and see whether the outcome is synergistic or not. So this was an experiment done recently. You can see it's a large number of combinations, 100,000 combinations, and only 5% of them were synergistic. And this is in cell lines. So this is a preclinical research. And when you convert, when you go from preclinical to clinical, there's a huge rate of attrition because what works in lab may not work in a mice model, may not work in a patient, and so on, right? So only 5% of success rate in terms of whether any two drugs given together will lead to more cell death as compared to those drugs being given individually, that is actually very low. And mathematical models again have a role to play here to increase this rate by looking at the mechanisms more carefully and suggesting which combination is actually going to work. Next slide, please. Now, again, if you draw an analogy, if you imagine the scenario in which your car brakes worked in only 5% of the cases, and you did not know which 5% of those cases they were working, you would never feel comfortable driving such a car. But that is where we are when it comes to combination therapies as of today in the clinic. Next slide, please. So why is there such a huge difference in systems which are engineered systems versus if you compare it to any living system? And the major reasons are, because the living systems have evolved over multiple different challenges, which we even do not have a good catalog of. Right? The engineered systems have also evolved, but we have a very good catalog of why a change was made in response to what objective function and what were you trying to achieve and so on. And also for engineered system, you have a design manual available because we as species, as a human species, have designed these systems, but we have not designed different living systems. So we do not know what part is connected to what part and why is it connected there. That information is not as comprehensively known as for engineered systems. So in absence of which, fixing these real living systems actually becomes a challenging task. Next slide, please. So the concept of digital twin, for instance, is coming into, which is clearly used by engineering industry in many scenarios, is becoming slowly more popular in the field of biomedical research. So the concept here is that, as the name suggests, that you have a digital model of a real object, and you constantly keep collecting data how that object or that system is performing in reality, and you incorporate those changes in the digital model so that you could actually have a more accurate prediction of when the system is likely to misbehave and in what manner. And you could have that information before the event happens, and therefore you could preempt that scenario. That is the goal with which these tools are being used in the industry in a very regular manner when it comes to engineering scenarios. Next slide, please. And these themes, as I said, have begun to enter the field of oncology as well. So people are creating patient-level digital twin models, where again it's a very recent field, and experts from different domains, engineers, AI, ML scientists, of course, biologists, physiologists, oncologists, they're all working together to see if they could create as accurate a model of a cancer possible, so that can be used for clinical decision making. Next slide, please. And of course, the technology has also developed. You can actually get more experimental data in a dynamic manner, so you don't necessarily are constricted by just the first and last scenarios. You could actually do liquid biopsy, which is a blood-based test. You could look at how many cancer cells are present in a given patient at a given point of time, instead of doing solid biopsy, which is taking chunks of tissues from the same patient multiple times. Next slide, please. So clearly we have made a lot of progress in understanding the fundamentals that yes, there are multiple outcomes possible of the same drug in a given population. Of course, a large proportion of them are going to die, but some of them actually switch to a different state, which might be more aggressive, or some of them switch to another state, which is more dormant. And there are examples of this which have also been shown. But this is all understanding at a single cell level, what's going on in a population. How are these numbers decided? How many cells do option A, B, or C? And how does that number depend on dose of the drug, timing of the drug, patient, individual patient scenarios? All of those are exciting open questions that we now have the ability to tackle. Next slide, please. So there have been also various initiatives, interdisciplinary initiatives. Next slide, please. And if the sort of the general point I'm trying to say here is that if cancer cells can acquire different identities in response to different therapies or different stresses, then we as a community who have different disciplinary identities can also crosstalk and learn from each other, communicate with each other, collaborate with each other to get a better handle of what exactly is going on and how to eventually impact the patient outcome in the clinic. Next slide, please. And there are various initiatives in this area supported by different governments all across the world. So there's National Cancer Institute in the US, which has a scheme of funding where they actually get together people working on mathematical modeling as well as experiments and different biophysical interactions. There's a very active mathematical oncology subgroup of the Society for Mathematical Biology, which is the most active international community in this area. Recently at IISc, we have started a center called Paramhansa Center for Computational Oncology, whose goal is to do training and capacity building in this area, and we'll be conducting many such workshops, 2 to 3 day workshops or even longer workshops in different parts of the country to actually bring together communities, bring together experts from different disciplines, and seed and nurture such collaborations with a clear translational goal in mind. Next slide, please. So in general, the field of cancer has been thinking about these so-called hallmarks of cancer, which are clearly many of them are more static in nature, and the request here is to begin to think of these hallmarks in a dynamic sense and identify what assumptions we have actually, hidden assumptions that we have been taking about the system. And again, math models have a crucial role to play here because they have been shown across different fields to understanding the fundamentals of different complex systems based on which we can actually make much accurate predictions these days. Next slide, please. So with that, I would just like to thank the fantastic set of students and colleagues who have had the fortune to work with, who again come from different disciplines, different trainings, and enrich our understanding, and also like to thank the funding agencies who have supported our work over the years. And thank you once again, Dr. Vamsi and Dr. Darshan, for having me here. It's been a real pleasure and honor, and I'll be very happy to take questions. And sorry once again about the technical issues due to which I had to request Dr. Vamsi.
No, not at all, sir. Yes. Over to Dr. Darshan.
Yeah, thank you, Dr. Mohit, for this illuminating talk on the cancer progression. So I would like to ask the attendees and participants if there are any questions. I had seen some comments by Dr. Anil Kumar sir in the chat. Sir, you would like to add anything or
Firstly,
comment on
Mohit,
it?
it was a very enjoyable session. And as I just commented there, the tumor cells as well as the microorganisms, they have a similar behavior. If you are given a patient, you start with some empirical antibiotic, then you know what is the sensitivity, which organism or which organism. And organisms of the same species and genus will intercommunicate with each other. They will share plasmid-based resistance between them. And there is also an inter-organism crosstalk as well. Similarly, in cells, as you know, if you take the natural history of some cells, for example, if you take papillary carcinoma of thyroid, patient is unlikely to die of that because that is going to be there for say 10 years or more at least. And therefore, how the tumor biology evolves, and once you start giving different types of monoclonal antibodies, what sort of stimulation does it have in the so-called survival of say 1 or 2% which have got escape mechanisms is one which will be interesting. And that's the reason why I wrote there, bespoke chemotherapy or bespoke monoclonal antibodies would mean that today for that patient, for that tumor, though you may classify as an ER positive carcinoma of breast, will be different as the time progresses in that particular individual because of the host versus the cancer reaction and between the cancer cells in the same conglomerate. And therefore, it becomes very interesting that how you have treatment customized to that patient today will change with time. And if at all theoretically I have an ability to create monoclonal antibodies at a very short notice, I will now continue fighting with the tumor. As it evolves, my monoclonal antibody armamentarium also evolves, and that fight hopefully will mean that, you know, either the host or the cancer gives up, and let us hope cancer gives up.
Absolutely, Dr. And I think that that's an excellent point that you have made. That yes, clearly the dynamics is very similar in terms of whether you look at scenario A or scenario B. It's the concept of sort of co-evolution and also community behavior, and the aspect of monoclonal antibodies. I think that's certainly a fantastic point. And we have been looking at some also. The field is emerging in the field of oncology. Also, this something which has been studied in microorganisms for a long period of time, persisters, so drug-tolerant persisters, which are actually able to survive the tumor, survive the treatment, in that case antibiotics, and in this case different targeted therapies. And what similarities and differences between persisters at an individual level and at a population level is something which is becoming more being researched, and we are also working on that together with other colleagues. So absolutely, you're right that it's the principles of collective behavior and emergent properties seen in different biological systems also being manifested here. And monoclonal antibodies is a great example, because in that case, as you rightly pointed out, one can track the data in a much more precise longitudinal manner, and then keep adapting one strategy rather than developing target therapies every single time, and hope that this armament would actually lead to a win in the battle. So yeah, I think that's a great point. I'll be happy to discuss further and see what we could work together on.
There are a few more questions in the comments, Dr. Jolly. Could you see and answer?
There's a question my student Sai.
Yes. So there's a question by Sai. How does the metastasis bypass the regulation of cytokine D, G1S, P53, or tumor suppressor genes in general, and BRCA in breast cancer case? So that's a great point, Sai. Now in this case, we have so far not coupled cell cycle into our models, but that is the next direction we are taking. How is the cell cycle, which is where most of the molecules that you mentioned come in, connected? How are the different cell cycle stages connected to EMT? Is EMT leading to suppression in cell cycle? If yes, then what is the distribution? How many of them are in G1S, G2M, and so that model is still being built, and hopefully by the time we chat again, I'll have some answers.
Thank you, Dr. Mohit. I think there are no more questions. So we're towards the end of the session. I would like to take this opportunity to express our deep gratitude to Dr. Mohit for sharing his profound knowledge and his groundbreaking research with all of us. Sir, you have thrown light on how the cancer progression is being handled and how the interdisciplinary approaches can address one of the most difficult challenge in modern medicine. So we are very appreciative of the time and the effort you have taken and contribute to our learning. I would also like to thank our center for organizing this event and providing a platform for the attendees to participate and also get new insights and inspirations. Once again, thank you, Dr. Mohit, for this exceptional talk. We also look forward to you in your continued contributions in this field so that we can have another talk on the recent inventions, what you are working on. Thank you so much, Saira.
Thank you very much. Thank you very much, Dr. Darshan. Thank you.